Now coming to the next topic. Okay, so before the next topic, you have to remember that there are certain applied aspects of this cell cytoskeleton. Okay, there are certain drugs which act on this cell cytoskeleton. So this colchicine, right? Colchicine and this vinblastine, right? They act on microtubule and it prevents the assembly of microtubule. I told you already here that there will be polymerization of the more and more tubulin molecule, that is known as microtubular assembly. When the colchicine or vinblastine is given, it will attach with the microtubule and it will prevent this assembly of microtubule, because of which the cell death will occur, right? Similarly, if you look at the another anti-cancer drug, that is the paclitaxel, or commonly known as Taxol group of drugs, it also binds to microtubule and makes the microtubule so rigid that the disassembly of microtubule is not possible. Getting my point? So this colchicine and the vinblastine, they are preventing the assembly of the microtubule. And this paclitaxel, they attach to in microtubule and makes the microtubule so hard that disassembly of the microtubule is not possible. Now, if the disassembly of the microtubule is not possible, what will happen? It will not be able to form the mitotic spindle, and what will happen? Ultimately, the cell death is going to happen. So that's why this is used as an anti-cancer drug. It causes the cell death. So this is the applied aspect of microtubule that you have to remember. Now we are coming to the next topic, that is the intercellular junction. I told you intercellular junction means that is the cell to cell interaction, right? So suppose I am drawing one epithelium cell. Suppose this is one epithelium cell, right? Okay. This is the nucleus and epithelium cell. And I'm drawing another epithelium cell here. Another epithelium cell. This is the apical part. And this is the basal part. So question is, how these two epithelium cells remain attached with one another? And second question is that, how this epithelium cell remain attached with the basement membrane. So this is the basement membrane and extracellular matrix. Okay. So cell to cell interaction and cell basement membrane interaction. Cell extracellular matrix interaction. Right. That we are going to discuss here. So cell to cell interaction. Right. At the apical most portion of the cell, okay, two cells remain attached with one another with the help of certain protein. So if I magnify this, so this is certain protein from one cell. This is interacting with similar kind of protein from another cell, okay, or the nearby cell. And when they are making this interaction, this junction is known as tight junction. This junction is known as TJ, tight junction. So I am writing here tight junction. Okay, so the protein which is forming this tight junction, the protein which are interacting with one another from one cell, another cell, they are interacting, and this junction is known as tight junction. The protein which forms this tight junction is claudin, CLAUDIN, and occludin. Okay, so you must have to remember this name. Claudin and occludin, these are the protein, okay, which interact from one cell, okay, to the other cell, and it forms the tight junction, which is present at the apicalmost portion of the cell, at the top of the cell, tight junction, right? Just below this tight junction, there is another interaction, okay. So, certain protein from one cell is interacting with similar kind of protein from another cell and is making a junction. This is known as adherent junction. So this is known as adherent junction. Okay. Adherent junction is again formed by protein molecule which is known as cadherin and catenin. Cadherin and catenin. Getting my point? So adherent junction is formed by protein, two protein I have to remember, cadherin and catenin. That is adherent junction. Now adherent junction, it is present on membrane. One membrane, another membrane, interaction is adherent junction. Now adherent junction requires some support from intracellular portion also, so that this interaction becomes stronger. So adherent junction remain attached with certain cytoskeleton element, okay, inside the cell. So it is attached with certain cytoskeletal element inside the cell, and this cytoskeleton is nothing but microfilament or simply actin. Getting my point? So adherent junction is anchored or is attached with certain cell cytoskeleton, and that is actin. So A for adherent junction, A for actin. Remember like this, that adherent junction remain attached with actin intracellularly, right? Now below this adherent junction, okay, below this adherent junction, the third junction, so protein from one cell membrane interacting with similar kind of protein from another cell membrane. This is forming another junction. This junction is known as desmosome. Desmosome, right? So desmosome is formed by a protein which is known as desmogelin. Desmogelin. Okay. So desmosome is formed by a protein molecule which is known as desmogelin. You will be finding out this desmosome in myocardial cell membrane. It's a point where the mechanically two myocardium cells are mechanically attached with one another. This is the point which is known as desmosome, right? Now the desmosome remain connected with certain cytoskeleton element, and that is nothing but intermediate filament. So desmosome, similar to the adherent junction, it is also connected with certain cytoskeleton element, and that is nothing but intermediate filament, right? Now below this desmosome, there is a channel forming junction between cell to cell. Now so far we have discussed the junction through which no molecule can pass from one cell to other cell, like tight junction. Okay, molecule cannot pass from one cell to other cell, or adherent junction or desmosome. Now we are going to discuss another junction where there is a pore, there is a opening between one cell to other cell, okay, and this is like a simple channel. This is like a simple channel. So this is a junction. This junction is like a channel through which small molecule can pass from one cell to other cell. This channel, Okay, or this junction is known as gap junction. This is very, very important for MCQ. Gap junction. So the gap junction is formed by a protein molecule which is known as connection. We'll discuss this detail. The connection, connexin, what is the difference, that we'll discuss. But for the timing, remember, gap junction is like a channel which is formed by a protein which is known as connection. The internal diameter of this gap junction is 0.8 to 1.4 nanometer. 0.8 to 1.4 nanometer. This is very, very important for MCQ. 0.8 to 1.4 nanometer. Because of this internal pore, what will happen? The small molecule, like for example certain electrolytes, sodium, potassium, calcium, okay, or water, they can be transported from one cell to other cell, like for example water, okay, like for example sodium, like for example potassium, they can move from one cell to other cell. But please remember, gap junction is impermeable to protein molecule because they are larger in size. So that's a small molecule, water, sodium, potassium, they can pass through from one cell to other cell through this gap junction, but not the protein molecule. Okay, so this is all about cell to cell interaction. Now, if we look at cell to basement membrane interaction, then you have to remember there is one junction which is known as hemidesmosome. So this is known as hemidesmosome, right? Hemidesmosome is formed by a protein which belongs to integrin family of protein. Integrin family of proteins forms hemidesmosome, and hemidesmosome connected internally or intracellularly with intermediate filament like desmosome. So desmosome, hemidesmosome both are connected with intermediate filament intracellularly. Okay, so there will be hemidesmosome here also, hemidesmosome. Now apart from hemidesmosome, there is another one, and that is known as focal adhesion molecule. That is known as focal adhesion, right? Focal adhesion. Again, this focal adhesions forms by protein which belongs to integrin family of protein. Focal adhesion remain connected intracellularly with cytoskeleton element, and that is nothing but microfilament. So this is focal adhesion. Focal adhesion remain attached with microfilament intracellularly. This focal adhesion is a labile structure. It helps in movement of the cell from one part to other cell. So focal adhesion is an attachment point which is labile, which is not fixed, which helps in movement of the cell from one part to other part. So this is all about different intracellular junction, okay, intracellular junction which are attaching cell to cell as well as cell to basement membrane. Now I told you that this gap junction is very, very important for your MCQ. So you have to remember certain more point about gap junction. Okay. First, you look at this diagram which is given in Gyan. Okay. So I'm magnifying this diagram. Look at here. So what you're finding out, this is one plasma membrane. Okay. This is one membrane. And this is another plasma membrane from a nearby cell. Okay, getting my point? So two cell membrane are coming near to each other. But the area where the gap junction is formed, the plasma membrane become more closer. Okay, normally the distance between these two plasma membrane is 20 nanometer as it is given here, right? So this one. So this distance between two plasma membrane is 20 nanometer. But the area where this gap junction is formed, here two plasma membrane comes more closer to one another. That's why you are finding out in this diagram from Gyan that the area where this gap junction is present, these membranes are coming close to one another. And the distance between two membranes at the area where the gap junction is formed is 3.5 nanometer. 3.5 is not the diameter or not the related to the gap junction. It is the distance between two membranes where the gap junction is formed. Okay. Now you look at the gap junction. Okay. So you will be finding out that the gap junction is formed by two parts. This is one part on one cell membrane. This is another part on another cell membrane. That means if I magnify this, you are finding out this is one part on one cell membrane and this is another part on another cell membrane. So these two parts, when they are connected with one another, that is a full gap junction. Okay. And I told you the protein which forms this gap junction is connection. So this is one hemiconnection and this is another hemiconnection. So basically I can understand that connection which is forming the gap junction, it is composed of two hemiconnection molecule, okay? So one hemiconnection, another hemiconnection, they are forming a complete gap junction. Now, if I look at this hemiconnection, one of these hemiconnection, so if I look at one of these connection, I'll be finding out that one connection is equal to 6 subunits. So this is one subunit, okay? This is 2, this is 3, this is 4, this is 5, this is 6. So what I'm saying, that connection or hemiconnection, it is composed of 6 subunits. So one connection is equal to 6 connection molecule. 6 connection molecule, okay? And 2 hemiconnection forms a gap junction, right? And one of this connection or one of this hemiconnection is composed of 6 connection molecule, okay? So connection is a subunit of connection, okay? Now if you concentrate on a single connection molecule, so it is here, this is a single connection molecule. What you are finding out? This single connection molecule is composed of 1, 2, 3, 4 membrane spanning domain, as you can find out here. Number 1, number 2, number 3, number 4. They are spanning the membrane through and through. This, then this, then this, and then this. So that's why it is said that this hemiconnection is composed of connection. Connection is composed of 4 membrane spanning domain. Getting my point? So here it is written. Each of the 6 connection has 4 membrane spanning region. So this connection, okay, they consist of, each of this connection is consist of 4 membrane spanning region. This is the complete structure of gap junction that you have to remember, right? Now gap junction is very, very important molecule because, you know, Key gap junction is present in the myocardial cell. This is also present in other types of cells. And if there is mutation of this connection protein, okay, then gap junction will be dysfunctional. Okay, cell to cell interaction will not be there, and because of which many diseases are there, which is due to mutation in the connexin protein. So if there is mutation in connexin protein, there are multiple diseases. Altogether 20 different types of human disease has been identified so far. But you have to remember certain diseases, like for example Clouston syndrome, right? So this is basically a ectodermal dysplasia affecting the nails, hair, okay, skin, all this region. Then second is the erythrokeratoderma variabilis. Okay, migratory erythema developed over the skin. So again, this is related to skin disease, migratory erythema developed over skin. Then cataract, idiopathic atrial fibrillation, and X-linked Charcot-Marie-Tooth disease. All of you know that Charcot-Marie-Tooth disease is basically a disease of the peripheral nervous system along with muscle weakness. So here the subunit, different types of subunit, that which subunit of connexin is mutated is also given. So this is connexin 32, this is connexin 40, this is connexin 50 and 46. So this subunit you may remember, but this is not essential because it is very difficult to remember all of this subunit. But at least you remember that these are the diseases which originate due to mutation of the gap junction protein, the mutation of the connexin subunit of gap junction protein, right? So this is all about cell-cell interaction. So lastly, I will show you one diagram which is taken from molecular biology books. The cell-cell interaction that we have already discussed, everything is given here. So this is the topmost interaction between cell to cell, tight junction. Just below the tight junction, you are getting this, here's adherent junction. Below that, you are getting these desmosomes. And the combination of these three, tight junction, adherent junction, desmosome, they are known as junctional complex. Right? Below the junctional complex, you are finding out here, this is the gap junction, which is forming a channel, which is forming a pore from one cell to other cells. And at the basal level, okay, you are getting this hemidesmosome, and you are also getting actin-linked cell matrix interaction. This is nothing but focal adhesion, right? So this is all about cell to cell interaction and completion of cell physiology. Before completion, few MCQ you have to answer. So first one, plasma membrane is mainly composed of, look at this mainly, okay. So they are asking which has maximum percentage in plasma membrane. I told you repeatedly, although it is a lipid bilayer, protein percentage is maximum, that is 55%. So this is the answer. Shell shape and motility is provided by, okay, shell shape and motility. So cytoskeleton will be the answer. I can find out actin is also there, microtubule is also there, right? But shell shape and motility, the cilia, flagella, all of this thing, that is composed of microtubule, and the strongest cell cytoskeleton is microtubule, so better to choose microtubule in this question. The cell junction allowing exchange of cytoplasmic molecule between two cells, this is nothing but gap junction that we have discussed. So this is all about